Hi everyone, welcome back. Here we are in our second and probably final lecture for digestive. There might be one more after this, but we will see. So let's go ahead and start as always with our not really attendance questions. First, adding protons, H plus hydrogen ions, to something causes it to become more what? Next, is the pancreas an endocrine gland or an exocrine gland? And third, what are paracrine hormones? So go ahead and try to answer those. Hopefully you've done that, so let's see what the answers are. First, adding protons, H plus, hydrogen ions, to something causes it to become more what? Well, as H plus increases, remember that means pH decreases, so adding protons causes it to become more acidic by lowering the pH. Next, is the pancreas an endocrine or an exocrine gland? Well, it's going to be both. It is an endocrine gland because it produces hormones like insulin and glucagon, and it's also an exocrine gland because it produces digestive enzymes that it empties into the digestive tract. We've talked about the endocrine function already. We've briefly mentioned the exocrine function. Today we're going to look at those a little bit more. And lastly, what are paracrine hormones? Well, paracrine hormones are the hormones that, instead of acting on distant body tissues the way endocrine hormones typically do, well, paracrine hormones are released by a cell and they act on nearby cells and tissues. So all of this is going to come into play today. Let's go ahead and pick up where we left off last time. Previously, we had chewed some food, created a bolus, and swallowed that bolus, which traveled down the esophagus and got to the stomach. So now let's take a look at the stomach, beginning with microanatomy. So we still have all of those same layers that we saw previously in the alimentary canal histology, but instead of just having the two-layer muscularis externa, remember the longitudinal and the circular layers, well, the stomach has a lot more motility. It needs to be able to contract in different ways. So it actually has a third layer of the muscularis externa. It has something called the oblique layer, and oblique means kind of diagonally or at an angle, and this allows the stomach to kind of twist and contort a little bit more than if it only had the two layers. And this is important because this is where a lot of that mechanical breakdown occurs. The stomach is constantly squeezing and twisting so that it can really mix that substance that we just swallowed, that bolus. It's going to mix it up and combine it with some enzymes and some acid so that we can break it down into something like a paste. So here we see the stomach. Inside the stomach, there's it's not smooth. It's actually got these folds called rugae, and these have a couple of roles there. One, it creates more surface area. If there's more folded area inside, well, there's going to be more surface area. But also, it allows the stomach to stretch without causing damage. But here we've taken just a small section of the wall of the stomach and we've magnified it, and that's what we see here. And the wall of the stomach, it has these open pits. These are called gastric pits. And the gastric pits go downward and they're lined with simple columnar epithelial tissue. And the further we go down, there's going to be these very specialized cell types that produce the substances needed for digestion in the stomach. So, what type of cell types are found in those gastric pits? Well, that's what we see right here, and then a zoomed in area right here. Well, first we see something called mucous neck cells. And mucous neck cells, for the most part, they are just going to be the upper part of this gastric pit, or, well, technically the gastric gland, which is deeper down into the gastric pit. But then we get into something called parietal cells. Parietal cells are scattered around. We see them here in blue. And parietal cells, first they secrete hydrochloric acid, HCl. This is primarily what makes the stomach acid so, well, the stomach content so acidic. And intrinsic factor. Remember we talked about intrinsic factor back in blood. Intrinsic factor is produced by the stomach to allow it to absorb vitamin B12. We also have something called chief cells. There's quite a lot of chief cells, and chief cells produce some enzymes. They produce something called pepsinogen. And once pepsinogen is released, hydrochloric acid has created a pH in the stomach that causes the pepsinogen to be converted into an enzyme called pepsin. And pepsin is an enzyme that's going to start to break down proteins. We also have gastric lipase. Lipase is an enzyme that's going to start to break down fats and oils, lipids. So remember, we also produced lingual lipase in the mouth. So we have a couple of different lipases now that are breaking down fats and oils. We also have things called enteroendocrine cells, and these are pretty deep down near the bottom. And the enteroendocrine cells release histamine and serotonin. These are paracrine hormones. These act on nearby tissues. And somatostatin, which is an endocrine hormone, so it's going to act more distant. We also have something called gastrin being released, and gastrin actually regulates the release of all of these enzymes by the stomach. They also, if we go back, all of these being released are regulated by gastrin. It also regulates the mobility of the stomach, so how much the stomach is churning and how the stomach is going to empty. The inside lining of the stomach is protected by something called the mucosal barrier, because even though the stomach is producing acid, well, acid is still harmful to the stomach. So the mucosal barrier is going to protect the actual stomach lining from all of this digestive activity that's occurring inside. So what digestive processes occur in the stomach? Well, we have propulsion. The stomach is going to move this partially digested food stuff around. Well, one is going to move it out of the stomach when the time comes. But remember, in lab we learned that the stomach has different regions, And interestingly enough, as the stomach is digesting substance inside of it, it's moving those substances from one area to another and kind of pinching off here and there to create cavitations, to create regions where different things are happening within the stomach. So the stomach will use propulsion to move that partially digested foodstuff from one region to another region of the stomach. Obviously, we have mechanical digestion. Like I've said now a few times, the stomach is squeezing and twisting to break down those larger pieces of food into smaller pieces and ultimately into a paste called chyme. We have chemical digestion a little bit. More chemical digestion is actually going to occur in the next step, but we do have some chemical digestion at least beginning in the stomach. We're going to start to break down chemical bonds. There is a little bit of absorption that occurs in the stomach. So things that are lipid soluble, fats and oils, are going to be absorbed in the stomach, or at least some of them will be absorbed in the stomach. This includes alcohol. Alcohol is absorbed in the stomach. And aspirin. There are other drugs, but aspirin is the most common example of a drug that does get absorbed by the stomach. And as we said on the previous slide, intrinsic factor is produced by the stomach, which is needed to absorb vitamin B12. B12, remember, is important in red blood cell production. And if the stomach is damaged in some way, or in things like gastric surgeries, weight loss surgeries, where parts of the stomach are either removed or banded off so that they are not useful to the body, well, we can see things like low vitamin B12. This is pernicious anemia, remember that term. And in this case, typically what would happen is someone would get B12 injections. Otherwise, they could go into having low red blood cells and low oxygen, which is what pernicious anemia is. So how is gastric secretion regulated? Well, we have what is it that actually stimulates hydrochloric acid, HCl, and it's primarily regulated by the autonomic nervous system, acetylcholine, gastrin, which is released by the stomach itself, and histamine. So these three things will tell the stomach to release hydrochloric acid. And remember, hydrochloric acid, in addition to breaking down food, it will also stimulate pepsinogen to pepsin, which is needed for enzymatic activity. Also, interestingly enough, histamine, well, if someone has allergies, what do they take? They take antihistamines, like we talked about back in immune. And antihistamines will do things like dry up mucus in the respiratory tract. But it doesn't just target the respiratory tract. It lowers mucus secretion everywhere. It's not focused. So it's not super common, but it's not unheard of for someone who takes a lot of allergy medication to then also have digestive issues. Because, well, one, it can cause things like ulcers. If you don't have that mucus barrier in the stomach will start to digest itself a little bit. But histamine also is needed for stimulation of hydrochloric acid. So you could end up having digestive problems like not properly breaking your food down. Later, it can cause problems like extreme constipation. So histamine does play a role that's important in digestion. So what about different phases of digestion? So first, there's something called the cephalic phase, and this is prior to eating, this is kind of what we talked about a little bit in the last lecture. This is when you start to see food or smell food, or in some cases even think about food if someone's telling you about something that they ate that was really good. And this gets your body ready to eat. So during this phase, this activates the acetylcholine, the parasympathetic nervous system aspect of things. And you'll notice sometimes your stomach will start to growl and your mouth will start to water. This is all in preparation to start to eat. So the cephalic phase of gastric secretion is because of the acetylcholine telling your stomach, hey, let's start getting ready to break food down. You start to release hydrochloric acid even before food gets to your stomach, even before you have food in front of you sometimes. But then eventually you will eat and food will get to your stomach. So we enter something called the gastric phase. Food is now coming into your stomach. And this will do things like stretch your stomach. And we have stretch receptors in the wall of the stomach. And this is going to cause more of that secretory activity. And we are going to have different chemicals within the stomach from your food. So things like peptides, things like amino acids, basically small pieces that break off from proteins. These are also going to stimulate your stomach. And they do that by stimulating something called G cells. And G cells release gastrin. And gastrin travels into your blood and then eventually is going to start to stimulate even more digestive activity. But how do you inhibit this hydrochloric acid release? Well, when the stomach content gets really low, below pH 2, well, now we've got enough hydrochloric acid. We don't need more, so pH below 2 inhibits more hydrochloric acid to be released. And sympathetic nervous system activity, your fight or flight response. Remember, the parasympathetic PNS was rest and digest, and the sympathetic nervous system is the opposite of that. So when your sympathetic nervous system is activated, you will not release hydrochloric acid. A lot of your other digestive activities will slow down or stop because you need to get your body into fight mode. This is why people who have a lot of ongoing stress will end up having digestive issues because their sympathetic nervous system is staying activated, which suppresses the parasympathetic activity, which is needed for digestion. Okay, so now we're still talking about the stomach and release of those hydrochloric acid and digestive enzymes, but we're going to start to leave the stomach and go into the duodenum. So this is going to be called the intestinal phase. Remember, the duodenum or the duodenum is the first part of the small intestines, just beyond the pylorus of the stomach. So as food is, or well, now it's chyme, as chyme is ready to leave the stomach into the first part of the small intestines, there is still going to be some stuff that occurs in the stomach. So the intestinal phase. As soon as it's time to release some chyme into the duodenum, well, what's going to happen is a little bit of it is going to be released at a time. It's not the entire stomach contents gets dumped into the small intestines, a little bit at a time. And what happens is there's going to be release of something called intestinal gastrin. And intestinal gastrin or enteric gastrin is going to be released into the blood, and it's not going to be as strong of an effect as from the stomach, but here we're going to have some, more importantly, some inhibition of stomach secretion from the duodenum. So if we are ready to start releasing this chyme into the small intestines, well, really the stomach has pretty much done its job. We don't need to keep secreting that hydrochloric acid and those enzymes. So the duodenum is going to be telling the stomach, okay, let's stop with the release of everything. Let's start releasing the chyme into the small intestines. So as that chyme passes from the stomach into the small intestines, there's going to be some stretch on the duodenum. And that stretch or distension is going to feed back to the stomach to say, okay, we're releasing now, stop releasing your enzymes, open up a little bit, and start to release some chyme into the duodenum. Also, as the content starts to spill into the duodenum, there are some chemoreceptors that detect how acidic the chyme is, how fatty the chyme is, and if it is hypertonic. So still in the intestinal phase, there's going to be something called the enterogastric reflex. And the enterogastric reflex, again, is the duodenum feeding back and telling the stomach, hey, stop releasing those digestive enzymes. We're done. You are now releasing the chyme into the duodenum, so your job is finished. We have some enterogastrones. These are hormones. They're from the duodenum or the duodenum itself now. We've moved beyond the stomach. So here we are releasing hormones from enteroendocrine cells in the duodenum, this first part of the small intestines. And again, this more feeds back to the stomach and tells the stomach, stop secreting those enzymes and that hydrochloric acid. The stomach has done its job. It's now ready to empty into the duodenum. The duodenum is again saying to the stomach, hey, your job is done. Let me take it from here. So before we move on to the small intestines, let's still wrap up some stuff with the stomach. Let's talk about regulation of movements of the stomach. So as the stomach is filling, so you're swallowing, the bolus is getting to the stomach, we are filling the stomach up, and that activity, so the act of swallowing itself and the esophagus passing food into the stomach causes the stomach to relax. By receiving a bolus from the esophagus, the stomach begins to relax. This allows it to accommodate more bolus that will be coming so that it doesn't hurt itself by stretching. Instead, it relaxes. But this also triggers something called gastric accommodation. Now, think back to every time we've talked about muscle. When you stretch muscle, what does it do? Well, Frank Starling says when you stretch muscle, it responds by contracting. But the stomach is a little bit different. The smooth muscle in the stomach has this gastric accommodation that says when you stretch it, it actually relaxes a little bit more. So this allows the stomach to receive whatever content we decide to push into it by swallowing a meal. Then we have gastric contractile activity. So this is those 3 layers of smooth muscle that we talked about. The stomach can contract in essentially any direction. So it allows it to squeeze, it allows it to twist, it allows it to churn. And then we get to gastric emptying. So here we see right now the pyloric valve is closed, but we want to squeeze off and create just a little bit of a pocket. We could see here what I was talking about earlier. We're moving that chyme into the pylorus while separating from the rest of the stomach body. And this will allow the pyloric valve to open just a little bit, and we squeeze some of that chyme into the first part of the small intestine. And then it closes back so that we don't have too much going in. And during this, the stomach actually squeezes in the other direction, so we push some of that content, that chyme, back up into the upper parts of the body towards the fundus, towards the cardia. This prevents too much chyme from getting into the duodenum all at once. Well, sometimes food goes too far the other direction and we vomit. Well, the technical term for this is emesis or emesis, and there is a part of the medulla called the emetic center, and the emetic center gets your body ready to vomit. And normally when we vomit, it's because something that we have eaten is harmful. Maybe it's too spicy, maybe it's a little bit old and there's some bacteria in it, or whatever. Our body just decides, I should probably get rid of this. Time to project, and the emetic center is activated. Your mouth begins to water. This is to kind of coat your mouth so that that stomach acid that's coming up is not going to harm your mouth. You take a deep inspiration, getting ready to push, and your lower esophageal sphincter, well, that should be LEJ, lower esophageal junction, relaxes. And now the upper part of the stomach, the cardia, is going to have a pathway for that chyme to come up into the esophagus, and we have something called reverse peristalsis. And remember, peristalsis moves chyme from the upper part of the digestive tract to the lower part. Well, reverse peristalsis is the opposite of that. It's going to take stomach contents and move it into the esophagus and move it upward until it exits the body. Now, before we get to the intestines, let's look at some of those glands off to the side of the digestive tract that play a role in digestion. So, first, we're going to look at the liver. The liver is the largest gland in the body. It's the largest internal organ. That's what we see right here. It's got special blood flow. It's got the hepatic artery. The hepatic artery is going to be a branch coming off of the aorta. It's what's going to bring blood and nutrients and oxygen to the liver for the liver to use for itself. We have the hepatic vein, which is going to drain waste and carbon dioxide away from the liver, so blood going away from the liver. But then we have that hepatic portal vein that we talked about previously. The hepatic portal vein is what's bringing nutrient-laden food from the intestines, from the stomach. We also had the one branch coming from the spleen. So this is not bringing nutrients for the liver to use directly. This is bringing stuff to the liver for it to inspect. So if we look at the microanatomy of the liver, that's what we see right here. It's broken into these roughly hexagonal structures called lobules. So here we see one lobule, and liver cells make those lobules. These are called hepatocytes. So a liver cell is called a hepatocyte. Here we see kind of an artist representation of a lobule, and we can see lots of hepatocytes there. Running down the middle of a lobule is something called the central vein, and the central vein is what drains away going towards the hepatic vein to take blood away from the liver. We have something called a portal triad, and a portal triad we see here off to the sides. And the portal triad contains a branch of the hepatic artery. It's not the hepatic artery at this point, it's branched too many times, but it is just an artery, a branch of a portal vein, and a bile duct. So that's what we see right here. These three things make up a portal triad. We have the artery supplying nutrients to the lobule. We have a branch of the portal vein, which is going to be bringing blood from the intestines, from the stomach, from the spleen. And that portal vein is actually going to branch through the lobule itself. And then we have the bile duct, which is going to drain bile away that this lobule has produced. We have things called sinusoids running all through, and those sinusoids are kind of like, they're kind of veins, they are kind of capillaries, they're very open, so that as blood from the portal vein passes through, it's actually going to be exposed to all of these hepatocytes so that they can metabolize the substance that just came from our digestive system. We have special cells called stellate macrophages, and these are scattered throughout. These are going to also break down debris. Remember, macrophages, cells that eat things. And we have bile canaliculi. So after we have processed things like bilirubin, substances like that, it's going to pass into these bile canaliculi, which ultimately drain into the bile duct and then eventually towards the gallbladder. So the liver produces bile. It's going to pass into the gallbladder for storage, and then eventually it's going to pass into the duodenum. So here, if we trace these green areas right here, the gallbladder empties into the bile duct, which then passes down and empties into the small intestines at the duodenum. So what is bile? Well, bile contains bile salts and bilirubin, and bile salts are required for breaking down things like cholesterol and certain lipids. They're going to make those lipids more soluble in this aqueous environment that's kind of in the stomach, or in the chyme that's exiting the stomach. So we said that the bile is stored in the gallbladder, and that's its primary job, is just to hold bile until it's released. But sometimes, actually pretty commonly, the gallbladder fills up with things called gallstones, which we see right here. Gallstones often occur if the bile duct is blocked, or in certain situations where calcifications can occur throughout the body. Those can lead to gallstones. In someone who eats a lot of fatty food, that can cause gallstones. So it's really common for the gallbladder to be taken out. Moving on to another gland, talking about the pancreas. Previously, when we talked about the pancreas and endocrine, we were talking about those islets, the islets of Langerhans. We are now more concerned with the acini cells. So here we see those. This is called an acinus, and it is composed of the acini cells, or the acinar cells. This is the non-endocrine portion. It's actually the bulk of, it's about 98% of the pancreas. It's going to empty its secretion into a duct. So remember the pancreatic duct, and the pancreatic duct is now going to empty into the duodenum. So here we see the pancreatic ducts emptying into the first part of the small intestine. So what does the pancreas release? Well, it releases something that's called commonly pancreatic juice. But pancreatic juice actually has a lot of stuff going on. It's going to provide more enzymes than any other part of the digestive tract. It's going to provide proteases. These are different enzymes that break down different proteins. You don't need to know what each of these do, but some proteases that it produces are enteropeptidase, trypsin, carboxypeptidase, and chymotrypsin. So these are all proteases. These are all going to be involved in breaking down proteins. It also produces pancreatic amylase. So this is the third amylase that we've seen. The second amylase that we've seen. It's going to produce pancreatic lipase. This is the third lipase that we've seen. And it's going to produce nuclease. Remember, amylase is going to break down starches into simple sugars. Lipases are going to break down fats and oils. Nucleases are going to break down DNA and RNA. So all of the food that we eat is going to have some combination of proteins, carbohydrates, lipids, and nucleic acids, the four basic macromolecules. So the pancreas releases those enzymes into the duodenum. But remember, so did the gallbladder. So really what's happening internally is the gallbladder, and to a lesser extent the liver, is going to release bile into the bile duct. The pancreas is going to produce all of its enzymes into the pancreatic duct. And then the bile duct and the pancreatic duct come together into something called the hepatopancreatic ampulla, we see right here. And then that is going to empty into or join the duodenum here at the major duodenal papilla, this little opening right here. And it's all regulated by the hepatopancreatic sphincter. So these smooth muscles that are going to relax to allow those enzymes into the duodenum when chyme is being released from the stomach. And then it squeezes shut once that chyme has been passed on. So how do we regulate all of that? Well, it's mostly by the cholecystokinin that we talked about earlier and secretin. You don't need to know these specific activities. This is just to kind of show you how it's going to be from the duodenum, releasing substances into the blood, which then circulates to the pancreas and to the liver and gallbladder, so that it's a full feedback loop telling everything when to be released and when to stop being released. Now, I didn't mention it back here. I don't know, we might mention it a little bit later, but I do want to go ahead and mention it now. As this chyme is exiting the stomach, it's going to be very low pH. So another thing that the pancreas is going to release here is bicarbonate. So bicarbonate gets released into the duodenum with the chyme, and that's going to buffer that chyme so that it's not so acidic as it's going to pass into the rest of the small intestines. So food is now exiting the stomach in the form of chyme. It empties into the small intestines. So the small intestines, it's actually going to be three different regions. The first region, the duodenum or the duodenum, very short. This is the shortest region. It's about 10 to 12 inches. Then we pass into the jejunum. Then we pass into the ileum. And when we get to the ileum, there's going to be a valve, the ileocecal valve or the ileocecal sphincter. So as food is passing through the small intestines is actually where the vast majority of chemical digestion occurs. It's going to be the location where almost all of the absorption occurs. So here we see the small intestines. We cut a little bit away so that we could see inside. We see that it's folded up, and then each of those folds is folded up a little bit more. We take one of those smaller folds, expand it. That's what we see right here. And then along the surface of each of those, there are even more folds. So the small intestine is built in such a way that it has absolutely as much surface area as possible to accommodate all of that digestion and all of that absorption. So let's look at some of these features. First, these large folds are called plicae circulae. And if we were to look at an intestine, it's actually not a ring, it's a spiral kind of. And one of them will spiral not the entire length of the small intestines because they do end, as we see right here, but it will go quite a distance. And then there's another one that will kind of run alongside it. But these, again, are to increase surface area, and they also kind of keep a little bit of a pathway for the chyme to pass as it's moving from the duodenum toward that ileocecal juncture, or the ileocecal sphincter. Then along the surface, we can see that there are folds and folds and folds, and these are called villi. And the villi are projections of the small intestine that are lined with simple columnar epithelium. And the simple columnar epithelium, they are absorbing because they have those microvilli on top to increase surface area. So we have lots of absorption taking place. But within the villi, now we mentioned this briefly back in lymphatic. Inside of each of those villi is something called a lacteal. So we're absorbing a lot of substances from the chyme. We're getting the nutrients that we need. But remember, some of those nutrients are fats and oils. And if you think about lipids, they don't dissolve in blood. So we don't want them to get into the bloodstream. We don't want them to get into any of these capillaries that are in the villi. Instead, we want them to move into this specialized lymphatic vessel called a lacteal. So the fats and oils go into a lacteal. They are going to be bound to certain proteins before they are moved into the bloodstream. That allows them to remember the alpha and beta globulins. Well, they can carry things like fats and oils without it being a problem. Then we also have intestinal crypts, and those are these deep pits. They're kind of like the gastric pits in the stomach. And inside an intestinal crypt, we see one right here cut away. We have some specialized cell types. So let's look at some of the different cell types that we see along the villi and into the intestinal crypts. Well, most of them are just going to be absorbative, just basic epithelial cells that are there to absorb. Down in the crypts, we're going to have intestinal juice being produced. And this intestinal juice is going to help the the actual absorbative cells to absorb. We're going to have goblet cells along the entire villi. These are going to produce mucus, and remember, there's lots of, early on, it might be a little acidic. Later on, it's going to be pretty basic, but we want to line the intestinal wall with mucus to prevent those enzymes and those pH changes from doing any sort of damage. We have enteroendocrine cells, which are going to produce paracrine hormones to either talk to earlier tissues or later tissues, just tissues that are nearby, regulating these digestive activities. We have Paneth cells, which are down in those crypts. These are going to produce antimicrobial proteins to, just in case there's anything, well, one, in the chyme, but two, we also have a lot of bacteria in our intestines, all through our lower GI tract. And these antimicrobial kind of keep them in check because we have these bacteria and they're helpful because we keep them in check. Sometimes if we get sick, well, they could start to get higher in numbers. And if there's too many of them, then they're not so good anymore. So these antimicrobials can help keep them in check, make sure there's not too many of them. We have stem cells down deep in these intestinal crypts, and these start to make all of the other cell types that then travel up through the crypts. We also have Peyer's patches kind of scattered throughout the small intestines. We saw these back in lab when we were looking at the intestines in the microscope. Peyer's patches are a part of the lymphatic system. They're going to, everything that we're absorbing, they're going to kind of inspect to make sure that there's nothing that could harm us before it gets into our bloodstream. So now we've left the stomach, we've emptied from the liver, the gallbladder, the pancreas into the duodenum. We've mixed all of that with the chyme, and now the chyme is going to move on into the rest of the small intestines. So intestinal juice we've mentioned, kind of there to help make absorbing easier. There are going to be some enzymes in that intestinal juice, such as the bile that we just got from the gallbladder, digestive enzymes that we got from the stomach and from the pancreas, bicarbonate ions from the pancreas, and brush border enzymes, which are along the cell membrane. So all of these other enzymes were kind of squirting into the chyme, but brush border enzymes are along these folds, these microvilli. So they are not squirted into the chyme, they are actually along the wall of the intestines. But all of these enzymes are going to play a role in further breaking down that chyme, breaking chemical bonds, breaking them down into smaller and smaller and smaller molecules. So again, we saw how the duodenum regulates the entry of chyme from the stomach by telling the stomach when to release a little bit of chyme at a time and when to stop making those stomach-produced enzymes. So as the duodenum starts to fill with chyme, now we're going to start to move it through the rest of the small intestine. So how does motility and absorption get regulated? Well, we're going to see something called segmentation. We saw that drawn out earlier. Segmentation, we're going to take the paste and start to squeeze it into manageable little chunks. And then we're going to have something called a migrating motor complex. And this just It's kind of like if you looked at this occurring, it would look like a train moving through the intestines with individual boxcars. So ahead, the intestinal wall will dilate a little bit, and behind, it will constrict a little bit. And this is all regulated so that it occurs simultaneously. We are moving each of those little packets through the intestines with this migrating motor complex. And this is all regulated by that enteric nervous system and paracrine hormones. So as soon as you have finished a meal, a little ways after that, when your stomach begins to empty and the intestines begin to fill, we're going to move that chyme through the intestines, but it's going to move very slowly. It's going to move just a short distance at a time. But between meals, after you've eaten and it's been a bit and you haven't eaten again yet, well, the movement's going to be a little bit faster, and we're going to have waves occurring, pushing this chyme through the intestines in longer waves rather than shorter spurts. And the job of this is to clear the intestines. So we want to make sure there's no chyme kind of lingering in the intestines until eventually we get to the ileocecal valve. The ileocecal valve holds everything in the small intestines until it's time to move into the large intestines, at which point the ileocecal valve dilates. We see that right here. And soon the chyme will move into the large intestines. Now, by this point, the vast majority of water has already been reabsorbed. The nutrients have already been absorbed. We get to the large intestines, there's not a whole lot that's left to absorb. So let's talk about first what we see when we look at the large intestines, the gross anatomy, the things that we could see with the naked eye. Well, first there's something called Tinea coli, and the Tinea coli is what we see right here running this long kind of grayish structure. The Tinea coli is kind of a mix of smooth muscle and tendon. And then we have haustra, and haustra are these little pocket-like regions. We can see it looks like it's in segments. Each of those segments is a haustra. But the other thing, and if you come to see our cadaver, this is one of the things that's most noticeable. There are things kind of hanging from the wall on the outside of the large intestines called epiploic appendages. And these are essentially, they're just fat storage. So they hang from the wall of the large intestine. Just like the small intestine was divided into regions, well, the large intestine also divided into regions. The first area that chyme passes into is a pouch called the cecum. And extending from the cecum is a small finger-like projection called the appendix. The appendix, a lot of times you will hear say that it's vestigial, that it doesn't have a function, or we don't know its function or something like that. That is untrue. The the appendix does have a function. It actually houses lots of your beneficial gut bacteria. So when you get sick, sometimes that will kind of wipe out your gut bacteria. Or if you take antibiotics, antibiotics will kill various types of bacteria, the ones that are making you sick, but it will also kill your beneficial bacteria. So if you wipe out your gut bacteria, that can lead to things like digestive issues. It's common to get diarrhea if you've taken antibiotics or if you've just been very, very sick. So to repopulate those beneficial gut bacteria, well, they are released from the appendix to repopulate the good bacteria in your intestines. As that chyme empties into the large intestines and the cecum fills up, it moves into the first part here, going up, going towards, away from the small intestines. We're going to go into the colon, and the colon has different regions. We have the ascending colon, the transverse colon, the descending colon, and the sigmoid colon. Sigmoid just means S-shaped, so we can see that it's kind of twisted, almost like the letter S here. And as we are moving through the large intestines, we are absorbing, mostly we're just absorbing the remaining water. There's some vitamins that get absorbed in in the large intestine, but mostly we're just claiming the last little bit of water. We get to the end of the large intestines, and here we have the rectum. The rectum is just a storage site for feces. It doesn't actually do any reabsorption or anything like that. It's just a storage site. And then we have the anal canal, or the anus, which is where the feces will exit the body. So what else occurs in the large intestines? We just mentioned the bacteria that live in your intestines, and they are beneficial. We do need them. Well, because one, they ferment, which further breaks down some substances so that we can get a little bit more out of it. We have vitamin synthesis. The bacteria will take some of that chyme and metabolize it and produce vitamins, which then get absorbed by the large intestines. We talked about maintaining safety. We have to keep those from getting too populated, and that was some of the roles of those small intestine cells. We also have the role of the appendix, which we just mentioned. If those bacteria numbers drop, the appendix can repopulate. So, like I said, the large intestine, it's going to absorb the remaining water. There's not a whole lot of water left by this point. It's all been absorbed by the small intestines. But some of that remaining water is going to be reabsorbed. We're going to get the vitamins that those bacteria produce. We're going to absorb those. We're going to absorb electrolytes that are in that water still. We're going to propel that chyme, or what's left of that chyme, towards the rectum. And then we're going to remove everything that is left over. So it's nothing by this point that is useful to the body. And that's what feces is. It's just all of the waste product that was not useful to our body. At this point, we're going to remove it through the process of defecation. All right, so that brings us to the end of digestive. It brings us to the end of the module. And up next, we have our lecture exam. And I will talk to you in the next video.